Hello everyone, welcome to my channel Love of Biology. Today we are learning about division phase, that is the mitosis or M phase, which is also called as equal division. In animals, mitotic cell division is only seen in the diploid somatic cells, while in the plants, mitotic division can be seen in both haploid and the diploid cells. It is also called as equal division as the number of chromosomes in the parent and the progeny cells are same. So it is divided into karyokinesis and cytokinesis. Karyokinesis is the division of the nucleus, whereas the cytokinesis is division of the cell. So first let us see the karyokinesis. Karyokinesis is division of the nucleus. It is divided into four stages: prophase, metaphase, anaphase, and telophase. So these four stages we call it as karyokinesis. Let us discuss one by one. First one is the prophase. It follows the G2 phase of the interphase. In prophase, chromosomal material will condenses, that is, chromatin thread-like structure will condenses to form the compact chromosome. Initiation of the assembly of mitotic spindle with the help of microtubules and all the cell organelles like Golgi apparatus, endoplasmic reticulum, nucleolus, nuclear membrane, everything will disappears. Next, metaphase. Condensation of chromosome is completed and they can be observed clearly under the microscope in this stage. This is a stage at which morphology of the chromosomes is most easily studied. At this stage, metaphase chromosome is made up of two sister chromatids which are held together by the centromere in this phase, and they will move into the position at the center of the cell at the equator. The metaphase is characterized by all the chromosomes coming to lie at the center. You can see here in the metaphase they are lying at the center, so that's why we are calling this phase as a metaphase plate or equatorial plate because of their alignment of the chromosomes. Each chromosome connected by its kinetochore to spindle fibers from one pole, and its sister chromatid connected by its kinetochore to the spindle fiber from the opposite pole. So that's why they can lie at the equator because of the spindle fiber binding at kinetochore region. Next, anaphase. At the onset of the anaphase, each chromosome arranged at the metaphase plate is split simultaneously, and the two daughter chromatids begin to move towards the two poles. So the splitting of the centromere taking place, they move to the opposite poles, the separation. In this phase, we can see the different shapes of the chromosomes. Next, last phase of the karyokinesis, that is the telophase. At the beginning of telophase, the chromosome at their respective poles decondense and form the chromatin network. Nuclear envelope assembles and all the cell organelles will reappear. So this is quite opposite to the prophase because all the cell organelles are reappearing. Decondensation of the chromatin network to form the chromatin network, it is taking place, and the nuclear envelope, that is nuclear membrane, again it reappears in this phase. After that, cytokinesis. After karyokinesis, the cell itself divides into two daughter cells by a separate process. That is the cytokinesis. In animal cells, this is achieved by the appearance of a furrow in the plasma membrane. It gradually deepens. This furrow will gradually deepens and ultimately joins in the center, divides the cell cytoplasm into the two. Whereas in the plant cells, they will undergo cytokinesis by forming the cell plate at the center. The formation of the new cell wall begins with the formation of this cell plate that represent the middle lamella between the walls of two adjacent cells. At the time of cytoplasmic division, organelles like mitochondria, all these will be distributed between the two daughter cells. In some organisms, karyokinesis is not followed, as a result of which multinucleate condition arises, leading to the formation of syncytism. So this is about the cytokinesis. It will separate the one daughter cell into one parent cell into the two daughter cells, and this will continues if they wanted to divide. Otherwise, it will form the multinucleated condition. And the last importance of the mitosis. Mitosis results in the production of diploid daughter cells with identical genetic complement. Usually, the growth of multicellular organism due to the mitosis. Cell growth results in distributing the ratio between nucleus in the cytoplasm. Therefore, the cell will restore the ratio. Mitosis is important in cell repair. The cells of the upper layer of the epidermis, lining of the gut, blood cells are being constantly replaced. Mitotic divisions in the meristematic tissues, the apical and the lateral cambium, results in the continuous growth of the plants throughout their life. So it will continues. If any wound happens, so that will also be healed due to the mitosis. Thank you. Because of this, it will continue, the growth will continues. Here the prophase, everything you can see. Thank you for watching. Please subscribe my channel.